Hello, good afternoon.
Good
So
afternoon, ma'am.
today we will start with Eudogonium. Now from this chapter you can expect either 5 marks short note or 8 marks life cycle based question. That's all. There is not much 2-mark questions expected from here. Only like what is Macrandra species or what is Nanandra species, like this. 1 or 2 questions might come. Otherwise, this is meant for 5-mark questions and 8 or 10-mark questions. Okay. So let's start. So as we all know, we will start with systematic position of Eudogonium. See, Eudogonium is a free-living freshwater green algae that are both planktonic and benthic in nature. Benthic, planktonic you already know. Now, the thing is, what is benthic? Benthic, these are certain terms you should know because these terms are not only meant for botany and all, it should be also, I mean, you will come across these terms in environmental science, ecology, everywhere. The term benthic actually refers to anything that is associated with or occurring on the bottom of any water body. So animals and plants that live in or at the bottom of the water bodies are called benthos. Okay, so benthic means something. So benthic organisms are those organisms that are found at the bottom of the water body. Okay. Next point, they are unbranched filaments made up of multiple cylindrical cells arranged end to end. Now, come to the left top corner. Here you will see these cells. See, they are filamentous like a filament, and they have cylindrical cells. See, these cells are cylindrical and they are arranged end to end like this. Okay. Fine. Now we'll talk about the systematic position. It belongs to the division Chlorophyta, class Chlorophyceae, order Eudogoniales, family Eudogoniaceae, and genus Eudogonium. Systematic position is very important. You will have to learn it in order to get better marks. Okay. Now we will talk about its specialized cellular structure and the types of cells that are found in Eudogonium. So there are 2 types of specialized cells that exist. Apical cells and holdfast. Now come to this diagram. You will see apical cells means what? From the very word you can understand that these cells are present at the apex or at the tip of the filament. So where are they located? Located at the filament's tip. This cell is wider and rounder than the others. See, they are structurally or morphologically little different from the other with a distinct thickening in its cell wall. The apical cell is responsible for the filament's growth. So, if a question asks, name the cell responsible for the growth of the filament, your answer is the apical cell. Now, the next type of cell is which one? The holdfast, situated at the filament's base. So, apical cell is present at the apex part or the part of the filament, and the holdfast is present at the bottom part of the filament here. This colorless cell, it is colorless. This is a diagrammatic representation. So a color has been given to it just to distinguish it from the other cells. Okay, this is basically colorless. This colorless cell is elongated in nature. And it also has elongated outgrowths on its sides that aid in anchoring the filament to the surface. So what is this holdfast doing? See, this holdfast anchors the filament to the surface. Suppose this is a stone or a rock or a gravel. The filament is anchored on this stone surface by virtue of the holdfast. Next, see, each cell of the filament is made up of three layers. There are three layers. What are they? Cellulose layer, which is the innermost layer. Then comes the pectose layer, which is the middle layer, and the outermost layer is the chitin layer. This is a very important point. Very important point. This is very unique about this organism. It has a tri-layered cell wall, the innermost one being cellulosic, the middle one has pectose, and the outermost has chitin. The three layers give rigidity to the cells and also provide protection. Plasma membrane contains the protoplasm that houses a nucleus. So it is uninucleate, a nucleus. Reticulate chloroplast. Reticulate chloroplast means what? Net-like chloroplast. Reticulate means net-like. A central vacuole and cytoplasm. The cell sap, which is present inside the central vacuole, is composed of inorganic compounds, secretions, and excretions. So whatever these organisms are secreting or are excreting, these compounds are also found in the central vacuole. Okay. The nucleus is large and oval shaped and sits in the center of the cell. So you will find the nucleus, the large oval shaped nucleus at the center of the cell. Okay. The cells of Oedogonium also consist of typical Golgi apparatus and endoplasmic reticulum along with mitochondria. As we have earlier mentioned that it is an advanced eukaryotic cell, so it is evident that these organelles will be present in the cell of Oedogonium. Okay, so this is the morphology of Oedogonium. Now, if a question comes, then write a brief note on morphology of Oedogonium. What should you write? You will have to write these points. You will have to emphasize on these points. What are they? See, you will have to define what is an Oedogonium. That you need not write the systematic position for structure-based questions. You will have to draw a diagram of this type. And you will have to write about the specialized cell along with a description of each cell of the filament. These are the most important points which you are not going to miss out at any cost. I've made it very crisp and brief. Please don't make it even shorter. Right? Okay, now we will move on. See, there is nothing much in its reproduction, morphology. Next, we will talk about its reproductive features. The reproduction in Oedogonium occurs through both sexual as well as sexual means. That means you can see both sexual reproduction as well as asexual reproduction in Oedogonium. Okay, asexual reproduction happens through formation of zoospore. All right, zoospore is the most important word for asexual reproduction in Oedogonium. are released from specialized structure called sporangia. You can also call it zoosporangia. Okay, you can also call it zoosporangia. Okay, and sexual reproduction involves fusion of gametes to form a zygote. This is something we are familiar with, like N cells from male and female will fuse to form a 2N structure. This is something we are familiar with. This sexual reproduction is oogamous type. This is an advanced type of reproduction, oogamous reproduction, and can be monoecious or dioecious. The species may either be Macrandra species. Macrandra species are those species lacking the dwarf male, or Nanandra species. Nanandra species are those species which possess the dwarf male. So if in one word, you should remember Macrandrus means there is no dwarf males. Nanandrus means there are dwarf males. Dwarf males are dwarf from the very word you can understand. Dwarf means small. Dwarf males are small, short, antheridium producing filaments attached near the oogonia or the female sex organ. If you look at the asexual reproduction here, asexual and sexual reproduction is nicely shown in this diagram. In your exam, you need not draw all these structures. You can also only write these. Just writing these terms and putting arrows will also give you equal weightage. Just make it's like this to make it little prominent, or you can avoid that also. Just use these words and terms in your diagram. Right? In asexual reproduction, what is happening? This unbranched filament is there, which is producing or which is having that zoosporangium. This zoosporangium matures to produce the spores in it, or the zoospores in it. The zoospores, after maturation, they get liberated or they are released from the zoosporangium, and the zoosporangium germinates to give rise to a new filament. So here you can see there is no fusion of two two cells. There is no fusion seen here. But in sexual reproduction, what is happening? See, this filament or the plant body or the thallus has got two different sex organs. The ovum is produced in the oogonium, and antherozoids are produced in the male sex organ or antheridium. All right. Now the antherozoids as well as these antherozoids are liberated, right? And this ovum, this part is the female sex organ. This produces the oospore, which is initially 2N. This 2N undergoes reduction division, that is meiosis, and forms N. 2N undergoing reduction division means what? Undergoing meiosis, and it forms N cells. These N cells are nothing but zoospores. Two different things are seen here. The ovum here is N. Correct. Egg cell is haploid N. And the antherozoids are also N. These two N, see, these two are two different parts. See, I'm just coloring this arrow so that you understand better. This green arrow and this green arrow, these two shows you a different thing altogether. And this part is different. Ovum is N, antherozoids are N. two ends together, they will fuse to give rise to a two-end structure. This is one way. Again, when it produces oospore, this oospore undergoes meiosis to produce zoospore. These zoospores get liberated like it happened over here. The zoospores will grow into a new plant. So this is what is happening throughout the sexual cycle of Edagonium. Now see, now we will talk a little about this Macrandrus and Nanandrus species. In Macrandrus type, the Antheridium develops in the filament. The filament means any vegetative cell of the filament. See, here only you can see. See, here itself you can see that. This is the filament. Any cell in the filament it can develop. It's of two types, monoecious and dioecious. Macrandrus species or Macrandrus type can be of two types again. Okay, monoecious or dioecious. Monoecious means what? Homothallic or bisexual. That means both the sexes are present or born on the same filament. And dioecious means what? Heterothallic, that is unisexual. That means the two sexes are born on two different filaments. Okay, so let's see what's here in this type. Examples are also given: Edagonium fragile, Edagonium nodulosum, Edagonium hirni, all these. In this type, Antheridia and Eugonia are born on the same filament. In dioecious, what do you see? In this type, the Antheridia and Eugonia are born on different filaments. Clear? That is Macrandrus type. You don't need to elaborate on these topics. You just need to know what are these. That's all. Coming to Nanandrus species, in this type, the Antheridia and Eugonia are morphologically different. They are morphologically different. This type is always dioecious. Different means what? Heterothallic. And when heterothallic, it is always dioecious. The Antheridia are very small as compared to the Eugonia and are also known as dwarf males. The small male filaments attached to the female filaments near the Eugonium. This is how reproduction occurs. Let's see, coming to sexual reproduction, which I was just now discussing with you, showing you the picture. What happens here? See, sexual reproduction in Eudogonium is oogamous. So you see oogamous type of reproduction. What happens here? The species can be Macrandrus or Nanandrus. That is not the point. Wherever it is, you can see oogamous type of reproduction. Clear? Now, each consists of a single cell. They consist of single cell. Clear? The single cell is a cell here. We will have single egg. Antheridium may contain 1 to 2 multiflagellated sperm cells. 1 or 2 multiflagellated sperm cells. All right. The motile gametes released from the Antheridia are chemotactically attracted to the Eugonia. What is chemotactic attraction? Chemotactic attraction is some chemoattractants. That is, some chemicals are secreted. Okay, some chemicals are secreted in the water body itself, and from the sperm, or sorry, the antherozoids, they can perceive this chemoattractants and they are attracted towards the Eugonia. So in different other organism also it is seen that the Eugonia secretes, or the female cells secrete certain chemoattractants in the water body which helps to guide the male cells towards the female female cells or the egg cell. Okay. Now look at this picture nicely. This is an This is a picture which we just now said, smaller antheridial filament that gets attached to the, see, read this and then try to match it here. This is the oospore here, the unfertilized oogonia, all these things, and the smaller antheridial filament is found somewhere here. So now see, now we will see. Only one sperm cell enters the oogonia via a pore. Many sperm cells are released in the media, that is water body. Only one sperm cell enters the oogonia via a pore, and fertilization will occur. That is, for fertilization to take place, only one male cell is enough. The zygote divides by meiosis to form four multiflagellated cells. These cells find substratum individually and divide to form new filaments. The life cycle of Eudogonium is thus haplontic in nature. Now, what is haplontic life cycle is something you already know. That is the reason why I have done the different patterns of life cycle very early in our class. You have learnt about this before, isn't it? This is your haplon life cycle. Isn't it? What happens in this haplontic life cycle? See, the haploid phase is much bigger and longer than the diploid phase. The diploid phase is the diploid phase is the zygote phase only. Here you see it is nicely divided, but this is more accurate because the haploid phase lasts longer in duration. See, the spores are N. The spores gives rise to the gametophyte, which is N. Then gametogenesis occur. That means what? Male and female gametes are produced. That is also N. But when these male and female gametes are fusing with one another, they are forming the 2N zygote. The 2N zygote immediately forms spores undergoing via meiosis. So this diploid phase is short-lived. The zygote is the only member of the diploid phase. So this is something very important. Clear? Now, one additional information I would just like to give you is steps of cell division. That is, I have not seen questions from here, but just point-wise, I would like you to know the various steps of cell division in Eudogonium. That is a part of this morphology only, this part. I have not included in this main video because I will share the link of another video which already has that explanation. Okay, you just go through it. I haven't seen questions coming on steps of cell division, but why to take chances? Why just know it? Okay, I'll just tell you the various steps of cell division in begonium. Just in case there is a question anytime, you will be in a position to write. So what is this? Steps of cell division in Edogonium. Okay. The first step, what happens, see, the nucleus shifts. I'm writing it. The nucleus shifts from the periphery towards the center. From the periphery towards the center, and then it gradually moves towards the upper half of the cell. All right. Then what happens? Ring-like thickenings will start to develop towards that upper part of cell wall. Right? Next step, what will happen? Ring-like thickenings develop towards the upper part of the cell wall. Okay. Then the nucleus will undergo mitosis. That means two daughter. God, why is this last one come? Then the nucleus will undergo. So it will undergo mitosis. That means what? If this is the nucleus, it will form two identical ones, 2n, 2n. There is no reduction. It will undergo mitosis. That means two daughter nuclei are generated. Fine. Now, after karyokinesis, in the next step, what will happen? See, after karyokinesis, a row of microtubule develops, and you know the function of microtubule? It plays an important role in cell division, formation of spindle fibers and all. I'm not discussing those in details now, but you should know these terms. Huh? Develops and accumulates between the two daughter nuclei forming a layer. Here, so this is what is happening, and then next see in the next step what's happening. See, this is actually your this ring-like thickening. If suppose if the cell is like this, the cell is like this, the ring-like thickening will be somewhere here. On the two sides, it will somewhat. The fifth step, what is happening? The ring-like thickening gradually elongates and splits. I'm not writing the full word. This ring-like thickening, I'm just writing like this. A ring-like thickening gradually elongates. Okay, it will gradually elongate and splits the mother cell, splits the mother cell towards the apex, right? Towards the apex and forms a concave cylindrical structure. And what is happening? See, the ring material forms the cuticle of the upper daughter cell. The ring material forms the cuticle of the upper daughter cell. Right? And now the upper part of the ruptured mother wall will remain. The upper part of the ruptured mother wall will now remain attached to the anterior end of the new daughter cell as the cap. You have seen that picture, no? That apical cap. Now that is how the cap is formed. So our next step is what? The upper part of the ruptured mother cell or mother cell wall, you should write, remains attached to the anterior end of the new daughter cell as a, okay. So this is what is actually happening in the cell division of Ergonium. Concept is easy. Diagram also will be shared with you. In case there is a question, you should be in a position to answer it. And finally, what happens, the floating septum will then go up to the future cell that is at the top of the mother cell at the ruptured end, and it will become fixed over there, and it will later develop mature cross walls. Okay, so this is how the cells are seen to have been dividing in Ergonium, and new side walls will also develop between the cuticle and the plasma lemma of the upper cell, and thus it will form two cells. This is how the cell division process gets completed. So the diagram of this will be shared with you. Link will be given in the description box. Please click onto it and go through it once. This is all about our discussion today. I hope you have started solving the question bank. It is a complete picture of the different types of questions that are possible to appear in the question paper. Go through the chapters properly. Try to answer those questions at a specific time. Don't take ages to write, otherwise you will not be able to finish your paper on time. So please see that you finish your paper on time. Take those questions, practice diagrams, and practice writing those at the same time. Only learning, reading, and preparing will not help. Finally, you are going to write in your paper. You are not going to appear for Viva exams. Therefore, please have the habit of writing. Okay, so till then, signing off. We will meet in our next class with the next new topic. We'll let you know the timings of our class. Hope you have understood today's lesson. Till then, goodbye.
Thank you, ma'am.
Thank you so much.
Thank you, ma'am.
Thank, thank you.